Welcome everybody to our first June meeting of the 2022 year of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. I need to remind you that the meeting is live streamed on YouTube and recorded and archived and publicly available. Today we have a single speaker, Gary Ann, who's going to be talking to us about material that he published recently. And I need to give you our usual preamble. Tom Helikar is here today, co-leading with me. We're always here to take your suggestions and comments. Jim Silico and Lorenzo are also part of the leadership team trying to keep things moving. Lorenzo has just worked intensively to come up with a system for making it easier for you to suggest speakers, and we'll come back to that in a minute. We have our usual communication channels. Please do help us use them. We have this MSM working group Slack, which I don't think people are using very much. We have our Twitter feed, MSM Viral. We have the IMAG MSM wiki page. NIH likes it when we add papers and conferences and other materials. If you give a seminar on these topics that was broadcast and archived, you can put links there. We have a LinkedIn page which you can use to advertise for your work, for hiring, these kinds of things. And we have our YouTube channel. Please do use that MSM Viral Twitter account to help us distribute our information. Are there any announcements for today? Besides the fact, Lorenzo, why don't you share that invite, that suggestion form?
Yeah, I'm sharing again in the chat. There is a new online form that is very short and easy to fill to suggest speaker for this seminar series. So please feel free to fill as many as you want of this form, and we will try really our best to invite your suggested speaker, as well as usual, if you want to candidate yourself for some of your postdoc, PhD student, any from your group, please do, just by sending an email to myself or any of the other people in the organizing committee. Thank you very much.
Okay. The other announcement I have is from Reinhardt is that the immune digital twin subgroup that would normally meet tomorrow is canceled for this week. So there will be no immune digital twin subgroup this week. Okay, coming up, we have Lawrence Calzone from the Institute Curie next week. We still do not have anyone booked for the 16th of June. So if somebody would like to step up for the 16th of June, that would be great. On the 23rd of June, we have Fiona McFarlane, one of the people from Mark Chaplin's group at St. Andrews, who's worked on pyroptosis modeling. And that should be quite interesting. So please do help us fill the calendar. As Lorenzo said, we've just created this form that should make it easy. If everybody fills out the form and puts in one name at the end of the meeting, we'll have 17 more people to invite. Remember again to think broadly in terms of the kinds of people you might like to hear. So without more ado, we will hear Gary talk about his recent paper on what it will take to get through the valley of death, to get to true precision medicine.
All right. Well, thank you for inviting me to speak again. This is my third talk for this group. That plus all my paragraph length sentence rants during these meetings. Probably I have more verbal time in this thing than anybody else. So I appreciate this. Reinhard and James have always encouraged me to somehow write down all the things that I talk about and comments that I make. And this essentially is that. For those of you who don't know me, I'm a surgeon. I'm a trauma surgeon, clinician. And I've been doing computational biology for over 20 years now. And I think I have a bit of a different background and how I arrived at this particular domain from most of the people who are participating in this, mainly because I have no formal training in any of the things that I will be talking about. And part of that is that when I learn something new, I'm always trying to figure out why doesn't everyone do this, which means that I'm intrinsically looking for limitations of what a particular method happens to have to do the very specific thing that I want, which is to have better treatments for my patients. And that clinical mindset is something that underpins everything that I do, and this very meandering path that I've taken to where what I do now. So the proximate motivation for this particular paper has to do with my position as a specialty editor for the journal Frontiers in Systems Biology. And I am the specialty section editor for something called Translational Systems Biology and in silico trials. So the part of the reason that I wrote this particular specialty grand challenge is people are like, well, what's translational systems biology? They want to know what it is so they can determine whether or not you want to submit something to this particular section. In fact, it was originally supposed to be translational systems biology alone, and I had in silico trials because no one knows what translational systems biology is. So I'm going to talk a little bit about the basis of that. And how, to a certain degree, in the mouthful of a title, reflects this kind of tyranny of terminology that we are facing, and all the associated, let's call them political consequences that are associated with terminology and how that is used or misused, and what sorts of challenges that we face as a multi-scale modeling community. Right. So translational systems biology was motivated by the Zeruni era NIH roadmap in the early 2000s, which talked about translational roadmap, which wanted to take all the excellent basic science that was being done at the NIH and convert that into something that was more directly clinically applicable. That was around the time I'd been doing, the type of modeling that I do, which is mechanism-based, agent-based modeling at cellular, multicellular systems. And I said, wow, that sounds really great, because when I heard systems biology, I thought, well, it sounds like systems biology. And I realized people who were doing systems biology didn't think what I was doing was systems biology. And I thought, well, this has something to do with computation, but then the people who were doing computational biology didn't think that what I was doing was computational biology. And a colleague of mine, Yoram Vodovitz at University of Pittsburgh, who I met in 2003, we came up, we were looking for terms to more precisely identify what it is that we were doing. And we talked about it for a couple of years and then wrote two papers in 2008, one in a burn journal, because it was a burn application initially. That was one of the exemplars. And then this PLOS computational biology, which interested the idea of translational systems biology of inflammation. And then we wrote a book about all these sorts of things that came out in 2015. So this is a pretty old term that that lost the fitness battle in terms of describing what it is that we actually do, because there are lots of other terms that now people say, oh, it's this, or it's that, or whatever. So we'll talk about that a little bit. So one of the primary features of translational systems biology when it was conceived of was the idea of using dynamic computational mechanism-based modeling to accelerate hypothesis testing with the supposition that these models represented a computational representation of hypothesis structures that people arrived at, and that this would be used to accelerate the hypothesis testing component of the scientific cycle. So all of you have heard the old chestnut about correlation does not equal causation, and this was intended to close the scientific loop from all the methods that were used to being identify correlation, which are statistical methods, blah, blah, blah. You would come up with hypothesis structures about how things actually worked as a result of that particular analysis. But the throughput of testing those hypotheses was limited by real world experimental processes, which have their logistical and practical limitations. The translational component was, since my interest was being able to affect my patient's health, was to simulate clinical contests, which led us to talk about in silico clinical trials, which came out in a couple of papers in 2004 for both of our groups. And the idea here is that we would create virtual populations and then treat them as study populations and apply hypothetical interventions and see whether or not they actually worked in the context of a model that mechanistically represented a patient. And this was also important. Translational systems biology had kind of a middle out strategy where we would focus primarily on mostly cellular behavior and using these model representations as abstractions of a lot of underlying detail to be able to determine what is conserved across biology, so species or individuals, et cetera. So some of these things now sound familiar, right? Because the dynamic computational modeling, that's essentially what multi-scale mechanistic modeling is. We all do that nowadays. People talk about in silico clinical trials all the time. It is a little bit different than exactly this, but lots of people are doing that type of thing. And then this idea of abstraction to identify conserved functions. I've written about it theoretically, but that's essentially a specification for a digital twin. Right, so some of these concepts have now evolved into other terms, which is kind of what this talk is about. So this goes to my desire to more formally describe what I would like to achieve. So as a clinician, I want to be able to treat any patient I have at any time during their disease process. To me, that sounds like precision medicine, right? Personalized precision medicine. I have an individual patient. I have an individual patient at a particular point in their disease process. I want to precisely target what is wrong with that particular patient and take that patient from not being sick to not being sick. All right. So how can that be expressed axiomatically? What is the minimally sufficient number of statements that are required to relay that particular concept? Right, so these are the axioms that I came up with several years ago. Axiom number one, patient A is not the same as patient B. This makes common sense. This is personalization. Axiom two, patient A at time X is not the same as patient A at time Y, right? People evolve over the course of the disease. They are different, and that calls for different things. That seems commonsensical, and that's precision. Axiom three, I want to take care of patients, right? Meaning that the goal of all of this is to find some way to treat that particular patient in a way that takes them from being ill to not being ill. Meaning that prognosis and prediction, while fine and is okay, is only useful if I can do something about that, right? So that runs into a little bit of controversy because there's a whole bunch of stuff that emphasizes prediction, but there's no pathway to control from those particular approaches. And the last one has to do with any overall program to do this should be able to take care of any patient that's sick. right? And to a certain degree, I came up with these axioms because my intuitive impression of what these words mean in English is not what they mean when people talk about them when the title is used together in various fields, right? So precision oncology, as laudable as it is, has no answer for anyone for whom the drugs don't work, right? It is essentially optimization for existing therapies and tailoring those to particular diseases, states for particular people, which is good, but it leaves out all the people for whom things don't actually work for. So it has a problem with it. There are lots of ways to prognose and predict and identify who's going to get sick or what's going to happen. But unless you can control that, it doesn't help. And a great deal of emphasis on genetic testing and genetic predisposition doesn't account for the fact that the disease states often don't manifest at a genetic level, right? They are dynamic protein-mediated disease states in which the genetic underpinnings of those patients remains the same, whether they're sick or not, or during parts of their disease process. So this is an initial foray into addressing the tyranny of terminology, because what has happened is that there are words that make sense, right, from a standard lexical context in natural language. And yet when they are applied in biomedical contexts, they get squished down to mean something very different from what a layperson would say. Oh, I think precision medicine sounds cool. I will let my congressperson, support that bill. And then they find out it's not what they think it actually is. So that's why I always talk about these particular things to provide a label for what I consider to be what we really want to achieve, which is the goal of medicine. So why does this not work? Well, it's because of the valley of death. So my opinion, and this is just my opinion, is that the most impactful failure point in drug development is the transition from preclinical studies to clinical implementation, right? And this drives me crazy because you hear about technological advances over here, like AlphaFold, systems biology, right? Humanized experiments, et cetera, which are all fine, but they are not going to revolutionize drug development because they're not the problem. The problem is here, right? The problem is taking what is otherwise a bioplausible compound that works by everything prior to putting it into a patient population. And when you put it into a patient population, it doesn't work, right? And the failure rate at this point is like 99%. And it is incredibly cost inefficient because it's very expensive to do this. And to me, when I hear about technologies that are going to revolutionize the ability to get new drugs to us, they're not, because they don't address this. Okay. Now, I consider the value of death is applicable to new drugs, obviously, but also combinations of drugs that haven't been tried before. If you try and do these things in the classical clinical trial way, there's a combinatorial problem up the wazoo that you cannot do just by picking things. And it's been demonstrated not to work particularly well. And in the COVID era, it's also repurposing of drugs, which is kind of ironic because a lot of the drugs that were repurposed for COVID sepsis, cytokine storm, were drugs that were failed sepsis drugs back when they were originally developed, and then subsequently became effective for rheumatologic diseases. So, my opinion, this is the thing that we need to deal with in terms of technology development. So what are the causes of the valley of death? These are my two main reasons why this happens. One has to do with biological heterogeneity. In other words, the intrinsic variability associated with biological objects and the various forms that object can take that leads to a similar phenotype, right? So if you think about how evolution works, this is baked into how biological systems are, and that the space of possibility is so large that it cannot be adequately addressed with standard experimental and data-centric techniques. That's the denominator problem I talked about in my first talk in this MSM working group years ago, and I'll show you a slide in a second. And the second thing has to do with a formal representation of what we actually know. Biology is primarily descriptive. It has a legacy coming from zoology and botany, where basically people sailed around the world and they listed the whole set of phenotypic characteristics of things. And they said, this thing is this because it has these parts lists, and this thing is another thing because it has this other parts lists, right? To a certain degree, molecular biology adopted those particular components, right? In terms of like genetic expression, blah, 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 all this kind of stuff. And there was a loss in recognizing the biological systems are dynamical processes in which the mechanistic generation of behavior resulting from the interaction of those parts is intrinsic to biology and disease, right? So we have no way of formally representing what that relationship is and how is that conserved across species, if it is conserved across species, or across individuals, right? This goes to personalized medicine and digital twins. So this is a quick thing of the denominator problem. Wrote this paper for Bulletin Mathematical Biology several years ago. And the idea here is that the total state space of possibility for a particular biological object represents this irregularly shaped multidimensional cloud. And our current ways of interrogating what that cloud is are limited. So we can either sample from that cloud, but we're only sampling a very, very small subset of what is possible. And we can make no assumption of what the distribution of it is because we don't know what the boundary conditions are, right? You can't make assumptions of normal distributions because normal distributions don't hold for systems of interacting components. Or we use tightly constrained experimental objects that are specifically designed to reduce heterogeneity so they become interpretable, that represent very small slices of that possibility space, right? So this paper proposes that the way to approximate what this space looks like is you construct a multi-scale mechanism-based computational model similar to this, and then use the power of high-performance computing to explore what that possibility space of that particular object actually is, and then give you something that you can then operate over. So that is part of why, I think mechanistic modeling is important. Note that any sort of data-centric approach is limited by B, C1, or C2. You cannot do A. Right, so why the valley of death exists? Because our current biomedical research processes directly lead to it. Right, there's too much variation in a study population often, right? Because the primary reason that drugs don't work is because for lack of efficacy, and the signal to noise ratio is always present. And everyone complains about patient heterogeneity in terms of determining whether or not it's effective. So everyone wants to cone down what your study population is. But the way that you do that narrows the population intrinsically to reduces the power of the clinical trial and makes it less applicable when you actually release it into the wild, which is why phase 4 drugs fail. And because we have no formal way of determining what it is that's different within that particular study population, the means of narrowing that study population is ill-posed, right? So we are just making a guess based on some sort of descriptive but non-generative process about why I can justifiably say this group of patients behave similarly enough for me to treat them the same. Because of that, there's no process for useful failure in clinical trials, right? We can just say, oh, that didn't work, and then you recycle bullet points 1, 2, and 3. So this obviously doesn't work because we're not any better at getting drugs than we have been before. And the question is, can we learn from other disciplines that are, let's call them more real sciences as opposed to biology in many ways? Yeah, so what do other sciences do to solve these particular problems? They do engineering, right? That this is the term that is used to solving problems in a system. You engineer a solution to that system, and the process of engineering is you have a formal process of identifying solutions given a set of constraints of that particular system. And modern engineering uses math and formal representations of those systems in order to use simulation to explore what the optimal solutions are, right? This is done in every other discipline and is the basis of all technological advances for the last 75 plus years, right? So, why is this hard for biology? Well, you can do bioengineering for biological systems that can be reduced to a physical system for which you can construct, physics-based models. So that works actually fine. You can do hemodynamic modeling that way. You can do material science modeling that way. But when you look at these types of things, the interface between the physical object and the biology is one term, right? It's one lumped term. And being able to engineer what happens at that biology, we are not good at that at all. All right. And that's why I say it's hard for what makes biology biology, because what makes biology biology is not chemical reactions, because that's chemistry. It's the fact that those chemical reactions take place in the context of cells that interact with each other, that modify what those chemical reactions are, and the behavior is generated by the aggregate behavior of those cells, right? And we do not know how to represent that, right? So there are no first principles or natural laws that govern those particular behaviors. We don't have the sufficient math to describe what that actually is. Because of that, we lack, quote unquote, trustworthy formal representations. Almost everyone on this call has dealt with someone who is either an engineer or a physicist and says, how do you ground your models? You're just making it up, right? Where's your first principles? Blah, blah, blah. We've all heard it, right? And it is a stopping point, a cognitive stopping point for an entire groups of people who are like, I don't buy that, right? And I don't buy it because I don't trust why you did this. Now, that forgets the fact that, well, what else are you going to do? Right? Because the people who don't use computational modeling are making those same assumptions and then operating on it based on intuition without any formal representation of how they believe what it is. It's just intuition. So that is a barrier that we face when we want to say we want to apply engineering to biological systems. Reverse engineering is extraordinarily difficult. That's why the Valley of Death exists, because by the very nature of why biological systems can evolve in heterogeneous environments, there is path non-uniqueness to phenotypes, right, based on the generative mechanisms that are there. Because if there was path specificity, then biological systems would be so brittle, evolution would not work. Right. So the fact that we have redundancies and multiple paths to ostensibly the same phenotype gives the diversity of behavior that's possible, that allows for adaptation to a shift in that particular environment. Right. Standard experimental biology does not address this at all. Right. And this is an epistemic limit of our current methodology. And given the way that people do experimental biology, this is intended to be pejorative. It's essentially alchemy, where you have extremely competent people who put together these highly engineered systems in order to get reproducible results of a particular perturbation. The problem is there is no formal way of taking that particular output and extending it to anything else other than, oh, I think it might work in this particular circumstance, right? So this is the problem that we face in biology. This inability to understand what is challenging about biology leads to the, in my opinion, potential mistranslation, but popularly appealing movement of terms from one field to another field, right? Because people talk about engineering biology without understanding why you can't engineer biology the way you can engineer an aircraft engine, right? Just as precision or personalized medicine would mean something who just knew what the meaning of those words were, as opposed to what is actually done, digital twins suffers from this particular problem as well. I've ranted about this for years. I've given up on this particular rant because it's better to be part of the problem as opposed to fighting the particular problem. I've learned this from Reinhardt. All right, so let's talk about medical digital twins just for a second and see how it links to translational systems biology and how we can get that, how we can construct something like that that can address the valley of death. Right, so I'm a first principles kind of guy. So the guy who introduced the idea of a digital twin, this Greaves fellow, right, laid down these three particular components that are essentially the axioms that are associated with a digital twin. Is a data structure for the real world system. There is some process that links those data structures together to form dynamics because you're interested in seeing how that particular real world object can potentially evolve over time, which is inherently a trajectory, which is inherently dynamics. And you take this in silico analog that has these particular properties, and then you link it back to the real world through some sort of data connection so that it can be updated to how the real world object actually behaves, right? And that is what is required for a digital twin. So what are the deeper concepts behind those three axioms? One, that data structure is a common formal specification that is shared by and links individual examples of whatever class of object you're making a digital twin of, right? Because you're making a digital twin of an individual, right? But that twin is of a population of objects that have separate instances, right? So this is the specification component that is easy for engineered objects because obviously they're engineered objects and they were engineered based on the formal specification. So you already got that, right, in an industrial application. The goal of the digital twin is to examine the future state, which means that you have to deal with trajectories. Now, this is really tricky at some level because how do you actually link these things together? In the industrial applications, it's easy. You do a simulation because you have the object that you've created and you know how the thing actually works. Now, we actually have to make an inference about what that is, recognizing that it is epistemically incomplete, right? And that there is always uncertainty associated with the trustworthiness of that particular object, right? But the idea is that common specification allows individual instances of this to have different behaviors if you simulate that model going forward. So simulation is inherent to the use of a digital twin. Now, what you use to simulate, we can argue about that, but it is not just a series of non-connected data structures. There's some process of transitioning from one data structure to another structure, right? I call that a mechanism. So if you take the axioms of precision medicine and you squish them together with what's necessary for a medical digital twin, right, you come up with these sorts of things, right? So a medical digital twin of any individual means you have to make a medical digital twin of any individual, right? Because you don't want to leave anybody out. So that accounts for personalization. It accounts for, which is axiom number one, it accounts for trajectories, right? Someone's different at time A, time B. That's number two. And you want to be inclusive for everyone. So that's number four, right? Because you don't want to say, oh, I can't make a twin of you because I don't have the specification to be able to do that, right? You want to be able to specify for anybody. Because you want to cover everybody, they have to encompass heterogeneity, right? Which follows those same axioms. So you need to have some way of generating heterogeneity while conserving the underlying specification. Trajectories are implicit, talked about for a second, right? And they are in every other field, almost every other field, associated with mechanistic relationships between the components of the reference system. This paper by Wright and Davison, who talks about the difference between a model and a digital twin, explicitly says, be very, very cautious about using data-centric modes for doing this because they're inherently undersampled, sparse, and brittle. And we'll get to that in a second as well. The medical digital twin says, I want to be able to treat. Now, you can come up with medical digital twins that are good for prediction. That's fine. That's not what I'm interested in. I want to have a pathway towards developing control. And finally, since we don't have the same degree of confidence in our first principles or in our natural laws, we need to account for the fact that there's perpetual epistemic uncertainty with respect to the specifications. And therefore, as opposed to having a perfect object, all right, or even having a perfect object, we just need medical twins that are good enough. All right, so therefore, finding a way to identify good enough needs to be an overall goal, which is axiom 4, right? So, in my opinion, a ideal program for developing medical digital twins interfaces with the axioms of precision medicine and has these particular properties associated with it. So, complex problems call for complex solutions. By the very nature of the problems that we face in disease right now, these are complex processes. And we have developed a set of very powerful tools that have been used to deal with complexity, namely, or untangle complexity, machine learning and AI. So, brief diversion here, because all of you know who've talked to me before, this is like one of the hurdles. And this goes back to my history of looking for why a particular solution is not perfect. And there are 3 things that everyone should know about ML and AI that comes from the ML and AI community that almost nobody ever talks about in biomedical ML and AI, though they fall into 3 groups. They either don't know about it, they know about it, but don't think it's applicable, or they're obfuscating the fact that these things exist. So the first one is that modern ML is essentially training artificial neural networks, and that artificial neural networks are governed by the universal approximation theory. They can approximate any function given any data set, right? That means that standard data splits, right, of training and testing data for internal validation is meaningless because you always get that. And that because of this, brittleness and underspecification are endemic problems associated with ML systems that the community understands, but it is only through failure in the biomedical community that it comes to light. And we're now seeing kind of the second wave of papers saying all these great image, algorithms, and they don't actually work so great, right? All these sepsis prediction algorithms or sepsis control algorithms, they don't work so great, right? And they don't work because of these 2 properties that everyone who does do ML and ANN should already know, but they don't communicate it to us because they're trying to get their thing applied. In addition to this, and this comes to the data sparseness and brittleness, is the curse of dimensionality associated with trying to identify the function, right? So this is a manifestation of the relative challenge of getting time series data to identify the trajectory in biology, and this countervening idea, which is a legacy of biology, which is just give me a more detailed description. So if you have 5 time points and 10,000 genes, you're going to fit, right? You're going to find an answer. It just doesn't mean anything because it's only applicable to that particular thing, right? So this, again, is a formal, provable constraint that is present that is somehow ignored by entire swaths of the biomedical community, right? You show me, RNA-seq data on 20 patients with, 5,000 genes that you're looking at, and you show me a line through a cloud of nonsense, and you're going to tell me that means something? It means nothing, right? So how does ML and AI deal with that? They deal with that by generating synthetic data for the things in which ML and AI works. So language and images. They haven't figured out how to do it for time series. And I'll get to that for a second, because this is really important and a particular leverage point for us. And the final one that's really important, that's less well known, but it should be well known, is the causal hierarchy theorem. Because while the universal approximation theorem says that any ANN can reconstitute any function, the causal hierarchy theorem says that ANN and the structural causal model that arrives from that ANN cannot predict the effect of an intervention on that function based on the data used to treat the ANN. What does this mean? What this means is you can't test anything novel, right? If you want to see what a new drug does, you can't do it, right? So you hear about people who are using ML or proposing use ML and AI to discover new drugs. You cannot do it. You can do it way far the front end of the Valley of Death pipeline, way before you get to the Valley of Death. But when the rubber hits the road, or actually the wheels hit the chasm of the Valley of Death, you can't do it, right? Because you have to run that clinical trial to get the data to see whether that thing was going to work. And if you have to do that to train your ML, then you've already done the thing that you're trying to make more efficient, right? So these three aspects should be present in every single paper that has to deal with ML and AI in biomedicine, right? These are the caveats related to any discussion about these particular things, why they're not applicable. So what's the exception to the causal hierarchy theorem? Simulation-based deep reinforcement learning. And it's an exception in quotes because it's not really an exception, because it isn't just relying on the training. It is the generation of synthetic data to provide the data in order to do the reinforcement learning. So most people interact with simulation-based DRL through the game playing AIs from DeepMind, and those are closed systems, and they work fine, and it's very interesting. And again, aside from the nonsensical hyperbole that's talked about in terms of other human or otherworldly or inhuman performance associated with these things, it's just a big lookup table that they've run billions of circumstances are, so they know what the probabilistic components of the decision tree are. It's not magic, right? It's an exception to the CHT because these things in aggregate are actually hybrid systems, because they incorporate mechanistic-based simulations, i.e. the rules of the game, to generate the synthetic data of the games that are played, so you can see what the counterfactual space is for a particular control strategy. Right. So what I would like to do is to say, this is how you find complex control solutions. I'm not going to put it on this slide, but I will assert that the biological processes that we are dealing with are too complex for classical optimal control strategies, because the models are too complex to be able to apply those particular methods to. Right. We had a whole workshop with Reinhardt about that. So the proposal is to use simulation-based DRL for complex control for discovery in biomedical problems. And we've done proof of concept work with respect to trying to control sepsis, for which there is no treatment. With these two papers, initially with Lawrence Livermore, in a simulation of sepsis using antibiotics, standard of care, and then with the advent of COVID, for which there's no effective antimicrobial, at least at the time, optimization of immunotherapy in that particular context. Right. So once you have established what sort of framework you want to be able to do your discovery in, now we move on to in silico trials, which are essentially populations of digital twins. And the redundancy in the title here is because we're not dealing with populations, which is a statistical epidemiological thing, right? And treats the entire glob as this distribution and then draws a line through it. The precision medicine component of this means that you have to account for all the different possible trajectories that individuals can take. right? And therefore, and because since that is the source of the failure of the end clinical trials, it is vital to encompass human heterogeneity with incomplete knowledge. All right, how do you determine something is good enough, given a potentially good enough specification? How do we actually do that in a way that produces the heterogeneity that you see in the clinical data sets? Because anyone who's looked at any sort of clinical data, it's all over the place. It's like a Jackson Pollock painting. All right. And that the idea is that formal specification is what captures what is similar between one particular instance of that system versus another particular instance of that system within the heterogeneity, but allows you to identify the specific counterfactuals that are associated with that particular instance. And it provides a way of characterizing the validity of your heterogeneity characterization, because it needs to encompass at least the distribution of data that's out there in the real world. So how do you do that? This is really challenging, right? So one approach, right, that is something that Chase Cockrell came up with, is the idea of using a different concept for what parameters are. I'll get to the issues of parameters later at the end of this rant, but right now, this is a different concept of what a parameter actually is. So if we think about how we actually represent a biological system, and we're just going to use an agent-based model as an example. You have this particular object, right, that has inputs and outputs. It has a series of, processes internal to it that are controlled by the molecular biology. It's all studied, produces some particular output. The actual set of functions that a cell can do are relatively limited, right? They're not that many things. They're probably like 7 or 8. How you get to that particular endpoint, the vast majority of the genes within that particular cell don't provide a new function, but essentially modulate that smaller subset of functions in some way, shape, or form. So if you construct your rule in a way such that the connectivity between components and the coefficients of those particular connectivity represents a different tuning or the gain, then the parameter space, expressed as a matrix associated with coefficients and interactions between all the components that you choose to list in your model, represents essentially a mathematical object that encompasses all the genetic, epigenetic, and environmental variability that's possible in that particular system, right? So I'm going to say that again. The idea is that you have this mathematical object that essentially encompasses all the various connections and weights of those connections between the components that you've chosen to represent in that model. And if you've chosen to represent in that model canonical things that incorporate the primary cellular functions that are present, right? So in sepsis, there's a pro-inflammatory set of pathways, there's anti-inflammatory set of pathways, there's apoptosis, there's migration, et cetera, right? There are certain molecules that are well established as the canonical molecules that are the mediators as the output, and everything else modulates those particular mediators. Therefore, if you construct a model in this particular fashion, you generate an MRM. Now you have a mathematical object that you can operate over using machine learning, genetic algorithms, and active learning, right? And if you take that approach and you fit it to that wideness of clinical data, right? So instead of trying to regress through it, you say, now all I'm looking for is a non-falsifiable configuration of the MRM, meaning that a stochastic replicate of that particular instance of the MRM is able to generate that data point. You can't tell me that that's invalid, right? Because you don't have any data to say that's invalid, right? And then you operate over that entire space, which is really, really large, right? But this is the strategy that you use to encompass heterogeneity, right? And this is in a paper. So what do we want to do at the end of all of this? What we want to do with translational systems biology, or whatever term you want to apply for this, you construct these synthetic population of digital twins with a way to encompass clinical heterogeneity. And then you apply AI, DRL control discovery in silico clinical trials, and you do plausibility testing, right? In an engineering paradigm that vastly increases the throughput that you have across the valley of death. Now, it's not going to replace a clinical trial because for the moment, these models are not trustworthy enough to substitute for patients for interventions. Doing it for control populations, maybe, right? But for the moment, because of the causal hierarchy theorem, you can't predispose from a data-centric standpoint what it's going to be. But this at least is a pathway towards applying engineering principles to the specific problem that is the primary barrier in getting us drugs that actually work. So, since it was Grand Challenge article for the journal, right, I posed these 4 grand challenges for people who are interested in doing this. How do you encompass and embrace the biological heterogeneity present in clinical populations in the design of silico clinical trials? Because too many attempts to create in silico populations are trying to wash that heterogeneity away, and you need to embrace it. And I presented one particular approach towards doing that. I'm not saying it's the only one, but I want other people to come up with other ways to do that. Number 2, this goes to how we can interface with machine learning. How do you generate synthetic data that obscures the generative simulation from the ML and AI approaches? Because of the universal approximation theorem, if you construct an ODE model, right, and just add a noise term to it, just as what someone would do for stochastic differential equations, the AI will see through that and recapitulate that underlying model, right? And it will become brittle because if that model is not applicable, then it won't work. So what you need to be able to do is create a noisy enough population of the appropriate types of biological noise such that you essentially obscure the generative model, because now you have A prime approaching A, which is in that denominator problem thing, okay? And the key here, and this is why I like agent-based models, is that stochasticity is applied in a biologically realistic fashion. So where does biological stochasticity exist? It exists at the gene regulatory level, right? Because you have a whole bunch of pathways converging down to a small number of genes, right? And Gillespie algorithms can deal with that. Spatial distributions, sometimes spatial things are important, right? Small nuances with respect to those particular configurations lead to divergent behaviors. Finally, the other thing is that a lot of these transition points, right, are stochastic in their nature, or at least in terms from an epistemic standpoint, we can treat them stochastically. If you generate a model with stochasticity embedded in those places, you will get more biologically realistic stochasticity, right? Because anyone who's looked at any sort of data set knows that the noise across different time points is not consistent from point A to point B, right? That consistency, that can vary. And therefore, a standard way of constructing a stochastic differential equation doesn't work for this. So the next challenge is how to design and use translational systems biologies, or we'll just call them MSMs, medical digital twins, to inform the next generation of clinical sensors and controllers. So this is important because this addresses the imperfection of the models that now. And I think it may be on my next slide, but the example is from weather prediction. So when in the 1950s, when the National Oceanic, NOAA, decided that we needed to have better ways of predicting the weather, and we use these mathematical models to do that. Of course, all the modelers said, well, the models aren't good enough. They said, that's okay. All right, you do it the best you can, and you use those models, just like models are used in physics, to define what do we sample? How do we look for things that can make your models better, improve your prediction, and provide iterative refinement? This is a huge gap we have in biomedicine because the biologists just pick what they think is right? And they use their intuition, and not to denigrate anyone's intuition, but it doesn't work very well, right? Or else we wouldn't have the problem that we have. So the idea here, in order to accomplish useful failure, is to use model-based information, model-based guidance of what do we sample, when do we sample, and then those present testable hypotheses that can be further invalidated based on sampling something new, right? And then this last one is how do we integrate mechanism-based translational systems biology or multi-scale modeling with developments in AI and machine learning to further control discovery and do this. So this is learning how to play well with others, right? Which we have already done, but other people don't do that, which is the next slide, right? So what are the grand challenges for us as a community? And I'm just going to say us as a community because we're in an MSM working group, so the MSM community, right? And I have said this multiple times before, there is an intrinsic bias based on some of the things I talked about before against the types of modeling that we do. And this type of bias falls into several classes, each one of which is a separate, a distinct, and sometimes contradictory strategy to try and overcome them. But all of you on this call, I have no doubt, have faced every one of these particular critiques from lots of people and represents probably the biggest headache in your life. So obviously, from a biologist side, they don't appreciate abstraction, right? They like, because the nature of biology is, I look at this particular detail, my expertise comes from this particular piece, that I know more about this particular piece than anybody else does, and this piece is different from that piece. So now that I have my expertise and this is my domain, right? If trying to link those things is antithetical to that particular mindset, right? And the fact of the matter is we need abstraction to determine what's similar and do engineering stuff. So if you want to solve the problem, this is the only way to solve the problem. From the physics side, we already talked about it a little bit. You don't have first principles, natural laws, mass conservation, energy conservation, blah, blah, blah. You don't have that kind of stuff, right? And if that's not in your model, then I don't believe it at all, right? Again, that stuff gets washed away because of statistical mechanics. Every time you do a chemical reaction model, you don't have to go back and do quantum states, right? At some level, this stuff ceases to become important. And the strategy is iterative refinement, right? We have a appropriately scaled complex model of sufficient complexity, right? That is linked hopefully to a data stream, digital twins, that allows for iterative refinement. This is one of the biggest barriers we have right now from the stats and ML side. We don't need you, we just need data. You give us data, we will give you the answer, right? This is a problem in current AI anyway. This is an ongoing debate for artificial general intelligence. Right, so this is really frustrating because people who do AI and ML should know what the limits of their methods are, and they kind of do, but they just can't do the things that are needed to accomplish the goals that we have. They just can't do it. So the strategy that I would try and apply to them is to say, we will help you be able to do the things that you want to be able to do, but you can't do given the constraints of the models. We won't say that part, by giving you what you have in every other field, which is synthetic data, to be able to learn more. And then we will kind of kill two birds with one stone. And voila, by the next thing you know, you have an integrated ML mechanistic model system, right? Which is what we actually want. This last one is extraordinarily frustrating and comes from the legacy of classical mathematical biology, which is that your models are too complex. There are too many parameters. You'll never parameterize it, blah, 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 right? Well, so on one hand, a model that's simple enough for you to do that is of no use to me is no use to me, so I don't care. And I think many people on the biology side who've dipped their toes in mathematical biology saw 3-equation tumor growth models with 100% efficacy of chemotherapy and said, this is nonsense. I'm not going to do this. It has nothing to do with what I do, which is a true statement, right? And that's true for a lot of models that form the standard, right, within our particular community, and to be perfectly blunt, sit on MABS review panels a lot. This goes back to what is a parameter? How do you ground a particular model? What is the validity of that particular parameter in the context that you have? So talk about this for a second. People want to ground things in vitro models. Fine, right? How do you know that the context of why that biochemical or movement parameter is generated in that in vitro system has any relationship to what happens in vivo? What's the temperature? What's the tension forces, whatever? You can measure all those sorts of things, but you never get that data at the in vitro level. I mean, in vivo level. They're not accessible parameters. You're never going to get that. So what do you do instead? Are you going to say, I'm not going to have a model that incorporates those things? That's self-defeating. We want a solution, right? We have to figure out a way to deal with that. So the MRM that I talked about, people don't buy it. I think it's sound, but this is a huge problem that we have, who those of us who deal with complex models face from people who should ostensibly be our allies, but they are not. Right, so one way around this is there an analog to the double descent of deep learning, right? Because the people who argue, the mathematical biologists who argue against complex, highly parameterized models, invoke my second most hated quote ever, which is the von Neumann thing about elements. I hate that quote. I hate that quote because it's a nonsensical quote. But it turns out that you can get around von Neumann's elephant if you have complex parameters, right? Which is essentially a multi-scale model, right? And this concept of double descent that's now prevalent in the ML community, right? Because they've got bajillion parameters on these things. And on one hand, they should be brittle, but they're not. So there might be some ways that we can pull in more formal explanations for why high complexity models should be used, other than the commonsensical thing that you should use a complex model for a complex problem, which is why this little, shrug emojis, I don't know what to do about that. I mean, at some point, the proof is in the pudding, and we're going to have to just do things and show that it's useful. Okay, so that is the rant. That is the talk. If I've said these things in certain circumstances, over and over again, but I'm happy to take any questions, and thank you for sitting through this. I'll stop the share if that's okay.
Okay, we have time for a few comments and questions. As always, I have some, but I know some people had to leave because it's already after 4. So I want to make sure that the people who have to leave sooner have a chance to discuss.
Well,
Don, go ahead.
Quang was going to ask something. I can wait.
yeah, the way to raise the hand.
I
Just
think
just unmute and ask the question. It's okay.
Okay, I have a question, Gary. This is Chang from Emory.
yeah.
So, I mean, I'm not a clinician. I have an MD degree, but I never practiced. So, I mean, the environmental health department here, what we do is more like an environmental chemical human health risk assessment. So, the population heterogeneity, in your case, individual heterogeneity, which is a kind of the big kind of hurdle for clinical translation. I think that's the same thing for kind of similar for human population health risk assessment, where we're dealing with the regulation, right? Like say you have to regulate the exposure to a certain chemical such that 95% of the population is protected, and then you can sacrifice the remaining 5%, what have you. which is the most susceptible, given a reasonable economic cost to control the exposure or remediate the pollution or have you. Here, in our case, traditionally we've been using animal-based toxicity testing to get some dose response curves, and starting from there, we're going to extrapolate to human population, and within that extrapolation, we have to consider these interspecies variabilities, as well as to consider what we call inter species, which means within human individual variabilities.
Individual variability.
Right. And then they do this using very, without any known additional information. They're doing this kind of uncertainty factors, like multiply by 10 for the interspecies extrapolation, then multiply by another 10 for inter-individual variabilities there. And those 10 can be broken down into like a 3.2 multiplied by 3.2. One account for the pharmacokinetics differences, and the other account for the pharmacodynamic differences. Yeah.
Where do those numbers come from?
I guess just some everybody agreed upon empirical numbers. I guess
so someone made those numbers up.
Yes. And then these days, in the last 10 years or so, we're pushing more animal-free, animal alternative testing by using just cell-based or organelle-based in vitro assays. Okay, and those tend to be using human cells or human origins, what have you, or maybe microfluidic system, organ on a chip. But even if we're using human tissue or human cells, we're still facing the same issue of how do you actually extrapolate to the risk for the whole population, where you're gonna demonstrate at this exposure level, like what percentage of population gonna have adverse effects, things like that. What are those response in terms of population risk? Yeah. So I guess we're facing similar issues, although in your case, you have to deal with individual patients to treat for them. Here, we don't have to really deal with individual, human beings. We deal with the entire population. Yes.
Yeah. So how I would approach that is you have some mechanistic change by which the toxicity at the cellular level manifests in some degree of organ dysfunction. Right. So you calibrate that the best you can with respect to the in vitro platforms that you actually have. And then you have some metric that's not necessarily concentration per se, but is a measurement of the cellular dysfunction that you're seeing that is potentially extractable from a clinical population for which you would then look for a disease process that involves those particular disorders. Because this is the key to the medical digital twin component. How do you do the twinning?
Mm-hmm.
Right? At what point in time do you say this individual is sufficiently twinned? If you just look at the parts lists, it's no good because it doesn't include the dynamics. So there is an inherent burn-in time in which you need to collect data to say that this particular real world person's trajectory is this, and this set of parameterizations for a specification form your allowable digital twins, because it's going to be an ensemble for that particular individual. So in your particular circumstance, you have some hypothesis of cellular dysfunction that's resulting from the toxin, right? You have a metric for what that cellular dysfunction is measured by something, right? Some mediator, something, right? And then you go look at a disease process that says, this is what goes on as a result of that particular cellular dysfunction. You find that clinical population and you say, what are the metrics in that particular population? And what is susceptibility to disease and disease trajectories in that particular population? So you're looking for an existent pathologic correlate to the consequences of the toxin that you're actually thinking about. Now, you can't do that unless you have a common specification that you can use to link all these things together, right? But when you think about what the toxic effects of a particular compound are, the toxicity by definition manifests at some sort of physiologic pathophysiology, right? There is a symptom complex associated with that. That symptom complex is the result of some degree of cellular organ level dysfunction. And I'll bet you there's a disease process that produces that particular dysfunction as well. You're not saying this is exactly the same, but you're saying that we now have a class of people with parameter space that have this susceptibility to that particular type of cellular dysfunction, and that will give you a more rationally identified population distribution within the community that might be susceptible to that particular toxin.
Yeah. I think one difference here for the risk assessment is we don't have the luxury as you guys, for clinical trial, to collect a lot of human data. Here with the chemical testing, or just heavily rely on either epidemiological probably measurements or some animal or cell-based assays.
So that's why your human population is an existing disease population with a similar dysfunction to what is produced by the toxin. So you're not giving the toxin to humans, that not doing human testing, right? But you're using the computational model to bridge what an imputed effect of the toxin is to some other pathological population for which someone is already studying anyway.
Yeah, usually the PK part is more manageable than the PD part in terms of heterogeneity.
Yeah, because that's easier.
Yeah, thanks.
As I was going to jump off, but I'll ask a question too, if you don't mind, Gary.
Go ahead.
So it's you don't see very many people like yourself, a practicing clinician, expanded, turned into a modeler. Do you, and since you've been doing mechanistic modeling for a while, do you find that you are able to translate the learnings that you've made over time through your mechanistic models in your practicing medicine too, in some complex, crazy complex cases?
The short answer is no. And the reason being is the goal of the modeling that I do is to get better therapeutic agents. And I don't have those agents. I say at the bedside, I wish I had this, but I don't have this. Mm-hmm. What actually has become the biggest insight that I have learned from all the modeling is essentially applying Popper and Karl Popper's falsification to clinical diagnosis, right? As opposed to attempting to verify a particular diagnosis that I have, I attempt to falsify the diagnosis that I have with the data streams that I am looking at, mm-hmm. right? Because my contention is that if you've made the right diagnosis and you're applying a particular protocol, then that's fine. You don't have to worry about it. Bad things happen when you're wrong. And therefore, you're constantly looking for circumstances in which you've made the wrong diagnosis, which means that you're looking for falsification, which means you have to be inherently skeptical about the hypothesis that you've made, right? Which should be science, but in current practice is often not. Mm-hmm. But in terms of so various insights with respect to sepsis that I have had, with respect to, some of the things that this model, which is nearly 20 years old, predicted is the preponderance of the anti-inflammatory component associated with nosocomial infections. And people don't recover because they're immune suppressed, and every system is damped down because you have an inappropriate negative inflammatory response to a non-evolutionarily identified perturbation, right? That's essentially what sepsis is. I can say I know this. I have no control to fix that. My argument against people who want to predict sepsis is like, okay, you have a predictive model that can tell me that 6 hours from now, this patient is going to get septic. Now what? What am I going to do about that? You have a predictive model that tells me this patient is going to need dialysis in 24 hours. Great. What do I do with that information? I have nothing to do with that information. There's nothing I can do with that.
You
Right.
make
So
sure
that's
you
why
have
there's
the,
an inherent component of control associated with what I want to be able to do.
Thank you.
Joel, did you have a question?
Yeah, I have a first of all, great talk. You brought up a lot of the questions that I think it permeates the mind of everyone that works with models. And I think one of the questions that I have, especially if we're looking on personalization and making it meaningful to an individual, is how we define what is healthy in the sense, of each person.
Well, so this goes to questions of surveillance, I suppose, and risk identification, which may be a little bit more along the lines of what Guang's trying to do, because you are now looking at essentially trying to establish a baseline, right? So at some level, the question is, well, how invasive do you need to get? What level do you need to have in order of identifying what is sufficient baseline? It very well may be that you don't need a mechanistic model to do that, right? That for instance, physiologic signal processing that's present in Fitbits and that type of stuff, they will tell you when you're about to get a cold, right? Because you have a change in your heart rate variability, and that tells you when you're about to get sick. There's an entire component of chronic diseases, health maintenance, all that kind of stuff. That is very interesting. Not what I do, because I only take care of them after they get sick. If they can avoid getting sick, that's fine, right? But the proximate challenge is once you seek out medical attention, we want to be able to do something for you when that actually happens. So surveillance, I think, can be accomplished through some of these more data-centric, predictive digital twins, like heart rate monitors, that type of stuff, et cetera. And then there's a light that goes off and says, you know what, you can get sick, right? You should go check something else, where now you're entering into a control phase in which you want to prevent something that's actually going to happen down the road. So I think that there's, certainly in the entire ecosystem of surveillance and health and non-health, you don't necessarily need a mechanistic representation of what health is. But this goes to the burn-in time I was talking about for the digital twin, is once you start getting sick, it needs to see how sick are you getting, right? So there's a whole potential trajectory space from that particular point. Where do you fall in that particular trajectory space?
Yeah, maybe a combination between the two, right? You have already a general model that it could be applied to the larger probability of the population. And then with whatever you're saying with is like a Fitbit or something that is telling you something's going wrong with you, you can estimate the point where this person is on this disease after you've had the first test coming in or something like that, right?
Yeah, I think that's a good strategy. So in a very grand sense, right, the concept of behind an immune digital twin, since the immune system, your inflammatory system, touches across a whole series of pathological disease processes, you would have some initialization period for identifying where in parameter space you happen to be. Once you identified whether or not you got sick. So a young, healthy person who gets in a car wreck, right, or gets a necrotizing soft tissue infection and gets septic from that, that patient is starting from a very different parameter space in terms of their responsiveness from a diabetic person who already has an elevated level of pro-inflammatory, levels, chronic inflammatory, increase, right, which is in itself a disease process, who gets pneumonia, right, and ends up in the ICU with something like that. So I think that, and this is part of the point of my last slide, I don't think anybody in this community thinks we don't need to have experimentalists, clinical scientists, and informaticians and machine learning AI people. all of us recognize the importance and the criticality of those particular capabilities and technologies towards this overall goal of making people better. Our problem is that they don't feel the same way about us, right? They feel like they can do it without us. And therefore, they're like, why do we have to have you participate in this? And it is even worse at a certain level, because in every other scientific discipline, science is driven by the people that do what we do, which is theory generation, model projection, model-directed experimental design, engineering principles, all that kind of stuff, right? That's how every other science works, except ours.
Yeah, thank you. Just to complement, I just watched a video of why people, car companies still spend a lot of money on those clay models before they actually build something.
Yeah. Mm-hmm.
And we are not doing the same thing, right? The community is not doing the same thing when the question is biology or medicine and stuff like that.
That's right.
Lorenzo, your question.
Hi. Well, I would say that I agree with most of the points you raised. Well, the general point, I agree with all of them. And about using experimental model, I don't think we need to lecture here. There is some value and usefulness. Perhaps the problem is you make some fun example like the beautiful cloud in some RNA-seq or omic stuff, or the in vitro that doesn't correspond to in vivo. These are models. As far as you know what you're doing, you circumstantiate your experiment and you can extract some valuable information. So it's important, to me, it's important to use computational model of theory just to guide where do we put the boundary to do the best experiment, so that this data then becomes valuable and give us a mechanism that we can encode in our ODEs for our digital twin or whatever. And that most of the time, I understand a little bit of the frustration might become from purely mechanistic experiments or papers that need to end up, and with these we discover a new mechanism that will cure cancer. It's more of the way things work. It's not really that the authors really believe that all the times.
Yeah, so part of that, and I agree completely with you, part of it has to do with where in the particular pipeline you want to expend that particular energy, right? So I think at a systems biology level, and that's why there's translational systems biology. In a classical systems biology, people understand the benefit of the models, right? Because they ground them in biochemistry, right? They have the high degree of resolution that's necessary to make people feel comfortable that those models are trustworthy enough to act upon, right? The challenge is when you translate upscales, right, out of experimental environments and deal with the messiness of the real world, right? So if you try and directly take those particular criteria that make you have trustworthiness of those particular methods, it's impossible, right? Because you can't get that kind of data in clinical populations of sufficient sample size to adequately represent what you actually want. Right, you have a very sparse sampling of what it is, and it's not a uniform distribution of what how those samples are connected. So I agree completely with you with respect to using computational models to enhance, laboratory, experimental, or even data analysis, sequencing type of approaches in order to generate hypotheses. But at some level, there's a hypothesis testing component that needs to be done. And that's the thing that's missing.
My last comment is that sometimes we have to make sure that to progress, we try to make hypotheses and then to falsify them in order to try to find a new one that is exciting, and we want to demonstrate it at all costs because we know that is impossible. So we better do the long way around, like theory says.
Well, so I think, again, using multi-scale models for high throughput hypothesis testing always leaves you with hypotheses that you can test some more, right? You're constraining the possibility space. or you realize your model is completely wrong and you find something completely new. Very rare does that ever happen, right? Because we kind of know the general level of how things actually work, right? There's really not that much fundamentally new that's being generated in any of this research that is being done. It's very much along the edges. The edges are important because that's where the heterogeneity comes from, but there's no way of linking that heterogeneity in any sort of formal way.
So I think formally we're out of time. I have a whole stack of questions that I have, but I can ask them when you come to visit. So
Okay, we'll talk about those.
But thank you, for a great talk.
Thanks for everyone's patience. My cat is like clawing my back. So,
Oh, well,
all right.
thank you, everybody. And we look forward to seeing you next week. Please send your suggestions for speakers. Thank you, Gary, again, for being so willing to put that together and walk us through your current state of thinking on these complex issues.
Thanks.
Okay, with that, I'll call the meeting to an end. Thank you, everyone.
Thank you, everyone.
Thank you.